Okay, so we've got some research here on this gene called SIRF1, and honestly, it's got me a little puzzled.
Yeah, SIRF1, it's definitely one of the more intriguing ones.
Well, it looks like it all comes down to mitochondria, those tiny powerhouses in our cells, and how this gene SIRF1 can really throw a wrench in their works.
You got it. Basically, SIRF1, it's a key player in building something called complex 4, and that's like the last stop on the energy production line inside the mitochondria.
All right, so complex 4, that's when the magic happens, where our cells get their fuel. But what happens if you mess with SIRF1?
Well, that's where things take a really interesting turn. You see, the scientists have created these knockout mice, and basically what that means is they've switched off the SIRF1 gene. Now, you'd think messing with energy production would have, pretty straightforward consequences, but that's not what we see.
Wait, you're saying these mice, with their energy production all messed up, they don't just, crash and burn?
Nope. Not only do they not crash and burn, they actually live longer than regular mice. It's a real paradox, and it's got scientists scratching their heads.
Hold on, longer lifespan with less efficient energy production. That just doesn't make sense. How's that even possible?
It's a puzzle for sure, but the research gives us some clues. It seems like in these knockout mice, their heart is the organ that really takes a hit.
So their hearts are working with a bit of a disadvantage then?
You could say that studies have shown a decrease in something called state 3 respiration in the hearts of these mice. Now, I know that sounds a bit technical, but just imagine your mitochondria are like these tiny factories churning out energy.
All right, I'm picturing it.
So state 3 respiration, it's basically a measure of how efficiently those factories are running at full speed. In these mice, the heart factories, well, they're working a little slower.
Gotcha. So less power output from the heart's mitochondria. But then how are these mice still going strong?
Well, they seem to have developed some pretty impressive compensatory mechanisms. It's like if your main power source is struggling, you got to find other ways to keep the lights on, right?
Right, that makes sense. So what are these mice doing to compensate?
One of the studies suggests that they might be upping their glucose uptake in the heart. It's like switching to a different fuel source to keep things running.
Interesting. So they're changing up their energy strategy. Anything else?
Oh, there's more. They're not just switching fuel sources, they're also increasing something called mitochondrial biogenesis, which basically means they're creating more mitochondria to make up for the fact that each individual one is less efficient.
So more power plants, even if each one is running at a lower capacity, it's pretty clever.
Right. And there's also evidence that something called the UPRMT is activated. Think of the UPRMT as like an emergency response team for the mitochondria. When things start to go wrong, the UPRMT kicks in, tries to fix the damage and protect the cell.
Okay, so these mice, they're like the ultimate survivors, figuring out ways to adapt and thrive, even with this SIRF1 deficiency.
Yeah.
What about other animals? I think you mentioned that this gene has been studied in other species too, right?
Absolutely. And here's where things get even more interesting, because the effects of messing with SIRF1, they weren't consistent across different species. In fact, in pigs, it's almost the opposite of what we see in mice.
Really? So switching off SIRF1 in pigs doesn't make them live longer?
Not even close. Actually, SIRF1 knockout pigs have a shorter lifespan than regular pigs.
Wow, there's a huge difference.
Yeah.
So what's happening in the pigs that makes them so vulnerable to this gene alteration?
Well, unlike the mice, where the effects seem to be mostly concentrated in the heart, the pigs experience a whole bunch of problems.
So it's not just about energy production then?
Right. The studies describe a pretty severe clinical picture. We're talking failure to thrive, muscle weakness, and even some facial abnormalities. It seems like their cells just aren't equipped to handle the disruption caused by the lack of SIRF1.
So no long-lived super pigs then?
Not quite. And when you look at their mitochondria, the situation is way more dire than in the mice. The pigs have multiple defects in their respiratory chain, which is the whole system responsible for energy production in the mitochondria.
So their entire energy system is compromised?
Pretty much. They have reduced activity of complexes I call in their skeletal muscle. So it's not just complex 4 that's affected. And there's also a decrease in something called citrate synthase activity, which points to more widespread mitochondrial dysfunction.
Whoa, this sounds a lot more serious than what we saw in the mice. It's like their cellular power grid is experiencing a complete blackout.
That's a great way to put it. And to make matters even worse, there's evidence that SIRF1 deficiency in pigs messes with the instructions for building these essential energy-producing complexes. It's like the blueprints are getting all scrambled, leading to unstable and inefficient power plants.
Okay, so we've got these two extremes. On one hand, you have the mice thriving with their SIRF1 deficiency, and on the other hand, you have the pigs struggling to survive. It's both fascinating and completely baffling. What could possibly explain this dramatic difference between species?
Well, that is the million-dollar question. It seems like there are many different layers to this puzzle. It's not just about how severe the mitochondrial dysfunction is, it's also about which tissues are most affected and how each species adapts to the challenge.
So the mice are kind of like those resourceful improvisers finding workarounds while the pigs are hitting a brick wall.
Exactly. It makes you wonder what's so different about their biology that leads to such opposite outcomes. Could it be their genetics, their metabolism, or something else entirely?
It's like we stumbled upon this fundamental mystery of life. Why do some organisms have more resilience than others? And what does that tell us about our own ability to adapt and age?
That's a question researchers are actively trying to answer, and the answers could have a profound impact on how we understand aging and longevity.
We've already uncovered so much, but there's another piece of this puzzle that we need to explore. What about those reactive oxygen species, or ROS? Where do they fit into all of this?
Ugh, ROS. The double-edged sword of cellular metabolism. You see, these reactive oxygen species are a natural byproduct of energy production in the mitochondria. They're like the exhaust fumes from our cellular power plants.
Right. I remember reading that too much ROS can damage cells and contribute to aging. But I also recall that they have some beneficial roles as well.
You're absolutely right. It's all about balance. A little ROS can be a good thing, acting as signaling molecules within the cell. But when they build up too much, they can cause havoc.
So how does SURF1 tie into this whole ROS situation?
Well, remember how SURF1 affects mitochondrial function? When you disrupt that delicate balance like we've seen in both the mice and the pigs, it inevitably has an impact on ROS production. The question is how much and in what direction?
So the less efficient power plants in the mice and the dysfunctional ones in the pigs, they could be leading to different levels of ROS. Is that what we're getting at?
Exactly. And it's quite possible that these altered ROS levels are contributing to the contrasting lifespan effects we see in these two species. It's another piece of the puzzle we need to figure out.
Yeah, it's a really intriguing possibility. Maybe in the mice, they're actually producing slightly less ROS, or maybe their antioxidant response is getting a boost somehow.
So their cells are experiencing a bit less wear and tear from those reactive oxygen species, maybe even getting some extra protection.
Exactly. That's the idea. And it actually lines up with some of the latest research suggesting that a little bit of mitochondrial stress, not too much, but not too little either, might actually be good for you in the long run.
It's like giving your cells a bit of a workout, pushing them to adapt and become more resilient.
That's a great analogy. And remember those compensatory mechanisms we talked about earlier? The increased mitochondrial biogenesis and the activation of the UPRMT in the SURF1 knockout mice? Those could actually be playing a role in this whole cellular resilience thing. Right? So it's like their cells are saying, hey, things aren't perfect, but we can handle this. But if a little stress is good, what about the pigs? It sounds like their ROS levels might be going in the opposite direction.
That's a valid concern. With their mitochondrial dysfunction being so severe, it's possible that their ROS production is going through the roof, overwhelming their cellular defenses and ultimately contributing to their shorter lifespan.
So it's like their power plants are pumping out way too much exhaust, and the cells just can't keep up with the cleanup.
Another great analogy. Of course, we need more research to really nail down the role of ROS in all of this, but it's definitely a crucial factor to consider when we're comparing the effects of SURF1 deficiency across different species.
It makes you wonder if there's a sort of Goldilocks zone for ROS, not too much, not too little, but just the right amount to promote health and longevity. And maybe that sweet spot varies between different animals.
That's an interesting thought. It really highlights the complexity of these systems. It's not just about one gene or one molecule. It's about this intricate interplay between genes, mitochondria, cellular stress responses, and yes, reactive oxygen species, all working together to influence lifespan and health span.
Okay, so we've got this intricate puzzle with pieces scattered across different species. Mice living longer, pigs facing a shortened lifespan. It's fascinating and mind-boggling all at the same time.
It really is. It's like nature is running this grand experiment, testing out different strategies for survival and longevity, and we as scientists, we're trying to figure out what it all means.
We've talked a lot about mice and pigs, but you mentioned earlier that SURF1 has been studied in other animals as well. What about those flies and worms? Where do they fit into this picture?
Oh yeah, I almost forgot about our tiny friends. The research on flies and worms is fascinating, although maybe not as extensive as the studies on mice and pigs, but it definitely offers some unique insights.
Okay, let's hear it. What happens when you mess with SURF1 in flies and worms? Do they defy the odds like the mice, or do they struggle like the pigs?
Well, it's not a simple answer. It seems like the effects of SURF1 deficiency in these creatures can really vary depending on the specific type of worm or fly and the conditions of the experiment.
So even more variation. This is getting
All
complicated.
right, but stick with me, it's worth exploring. In some studies with fruit flies, for example, reducing SIRF1 function led to a shorter lifespan, which kind of lines up with what we see in the pigs. It suggests that there might be some fundamental mechanisms that are conserved across these different species.
Okay, so in those flies, at least messing with SIRF1 has a negative impact on their lifespan.
Yes, but here's the twist. Other research with flies has actually shown that a mild reduction in SIRF1 activity can actually increase lifespan, especially when they're on a restricted diet.
Wait, hold on. So less SIRF1 can be bad or good for flies depending on what they're eating.
It seems that way, and it gets even more interesting. In a study with a type of nematode worm called C. elegans, knocking out WHIM, SIRF1 didn't really affect their lifespan under normal conditions, but when these worms were exposed to certain environmental stresses like heat shock, they actually lived longer than the normal worms.
Wow, so knocking out SIRF1 gives them an advantage when they're under stress. That's almost like those mice thriving despite their heart problems.
It is remarkable, isn't it? It suggests that SIRF1's role in longevity might be highly context dependent. It's not just about the gene itself, it's about how it interacts with other genetic and environmental factors.
Okay, my brain is officially doing backflips trying to keep up with all these different scenarios.
Right.
Mice living longer, pigs facing a shortened lifespan, flies with mixed results, worms getting a stress resistance boost. It's like every animal has its own unique response to this SIRF1 deficiency.
You got it. And that's what makes this research so exciting. It's challenging our assumptions about the relationship between energy production, cellular stress, and aging.
It seems like we're just scratching the surface of this whole complex and fascinating field. But in the midst of all this variation, are there any common threads that we can pull out? Any clues that might help us understand why SIRF1 has such diverse effects on lifespan?
That's a great question. And while the specific outcomes might differ, there are some recurring themes that we see popping up. For one, the research across all these different species points to the crucial role of mitochondria in aging. Whether it's the heart issues in the mice or the widespread mitochondrial dysfunction in the pigs, it's clear that these cellular powerhouses are at the center of the action.
Right. Disrupting their function even in subtle ways can have ripple effects throughout the entire organism.
Exactly. And another common thread is this idea of cellular adaptation. We see it in the mice with their compensatory mechanisms in the flies, adapting to dietary restriction, and even in the worms becoming more resistant to stress. It seems like cells have this incredible ability to adjust and respond to challenges.
So it's not just about the initial damage caused by messing with SIRF1. It's also about how well the cells can compensate for those changes.
Precisely. And this ability to adapt might actually be a key factor in determining whether a species benefits or suffers from SIRF1 deficiency.
It makes me wonder if there's a way to tap into those adaptive mechanisms, to somehow boost our own cells' ability to cope with stress and age gracefully.
No, that's a question that researchers are actively exploring. If we can understand how some organisms manage to thrive despite these mitochondrial challenges, maybe we can apply those insights to develop strategies for promoting healthy aging in humans.
Now, that's a thought-provoking idea. But before we get too carried away with future possibilities, let's take a step back and consider the broader implications of this research.
Good point. It's important to remember that these findings aren't just about mice and pigs and flies and worms. They're about understanding the fundamental processes that govern life and aging across the entire spectrum of living organisms.
It's like we're piecing together this grand tapestry of life, with each species offering its own unique thread to the overall pattern.
And the more we learn about these individual threads, the closer we get to understanding the intricate beauty and complexity of the tapestry as a whole.
Well said. We've covered a lot of ground in this deep dive, from the molecular intricacies of SIRF1 to the paradoxical effects on lifespan in different species. It's been a whirlwind tour of cellular power plants, reactive oxygen species, and the incredible adaptability of living organisms.
Yeah, it really makes you think that these tiny creatures, mice, pigs, flies, worms, they hold so many clues about the mysteries of aging. It's pretty amazing.
It is, right? You wouldn't think it, but they're giving us this incredible window into some really fundamental processes. And it all comes back to this. What can this research on SIRF1 and its impact on lifespan teach us about ourselves as humans?
Yeah, that's the big question, isn't it? I mean, we've got mice living longer with these less efficient power plants, pigs facing a shortened lifespan, flies with mixed results, worms getting this boost in stress resistance. It's a lot to wrap your head around. It is. But there are definitely some important takeaways here, even if we don't have all the answers yet. For one thing, this research really highlights just how crucial mitochondria are for our health and how long we live. It's not just about energy production, it's about cellular signaling, stress responses, this whole web of interconnected processes.
So those little power plants are even more important than we thought.
Absolutely. And, the fact that we see such different responses in different species, that really underscores the complexity of aging. It's not a one-size-fits-all kind of thing. There are all these different pathways, all these different factors at play, and what works for one species might not work for another.
Something else that really struck me is this theme of adaptation that we've seen throughout this whole deep dive. The mice finding workarounds for their heart issues, the flies adjusting to dietary restrictions, the worms becoming more resistant to stress. It's kind of inspiring in a way.
It is. It really speaks to the incredible resilience of living things. And it makes you wonder, can we harness that adaptive capacity? Can we learn from these creatures and figure out ways to boost our own resilience to the challenges of aging?
Now, that's a really interesting idea. Imagine if we could unlock the secrets of those long-lived mice, or figure out how to activate those stress-busting pathways in the worms. Could we potentially slow down aging or even prevent some of those age-related diseases?
It's definitely a possibility worth exploring. Of course, we're a long way from translating these findings into therapies for humans, but this research is giving us some valuable clues about the pathways and processes that we might be able to target if we want to enhance our health span, not just our lifespan.
Right. It's not just about living longer. It's about living well for longer.
Exactly. And understanding the role of genes like SIRF1 and how they impact mitochondrial function could be a crucial step in that direction.
Well, this deep dive has given me a lot to think about. I'm definitely looking at those little power plants in my cells with a whole new appreciation.
That's great to hear. And remember, it's not just about the science, it's about the wonder. The fact that we can even begin to unravel these mysteries is pretty incredible.
I couldn't agree more. So to our listener, we hope this exploration of SIRF1 and its impact on lifespan has left you feeling energized and curious to learn more.
And who knows, maybe you'll be the one to make the next big discovery in the field of aging. Keep those minds curious and those mitochondria humming.